Hello, my name is Robin De Leon, and I'm the Executive Director of Connecting Families Urea Cycle Disorders Foundation. This is an exciting time for families suffering with urea cycle disorders, as there are several upcoming clinical trials and possible treatments to further treat UCDs. Connecting Families will begin creating a series of videos this year, and one of those is collaborating with industry currently working towards treatments for UCDs. So many can be apprehensive about the risks of being involved in clinical trials, or even being placed on medications, and rightly so. These videos will be so that you can learn what is being offered and to decide for you and your family whether these treatment options may be something for you. Connecting Families UCD Foundation was invited by Arcturus Therapeutics for Rare Disease Day in 2022. We had the opportunity to share our UCD journey and represent urea cycle disorders. Please understand that these videos are being offered to you by us for educational and informational purposes only. Connecting Families UCD Foundation creates and produces these videos completely voluntarily, and we incur all costs in bringing these to you. We do this for you, so that you can learn about what's out there, and that so you can be more informed about some of the amazing treatments and options coming very soon. There will be links below to connect you to more information about the content, and if you would like to know more, please contact me with your questions either by email or phone at robin@ucdfamily.org or my number 918-490-3055, or you can use the Contact Us form on our website at www.ucdfamily.org. Thank you, and we hope you enjoy this video. Dr. David Geller is the Vice President of Pulmonary and Rare Diseases Clinical Development at Arcturus Therapeutics. Dr. Geller received his medical degree from the University of Iowa and trained in pediatrics and pulmonology at the University of California, Los Angeles, and the University of Arizona, respectively. Dr. Steven Cederbaum, Professor Emeritus of Psychiatry, Pediatrics, and Human Genetics. Dr. Cederbaum was born in Brooklyn, New York. He received his BS degree from Amherst College in 1959, majoring in chemistry. It was there that he developed his lifelong interest in biochemistry and genetics. He received his MD degree from New York University School of Medicine in 1964. He did residency training of internal medicine at Barnes Hospital, Washington University School of Medicine in St. Louis. He was trained in medical genetics at the University of Washington in Seattle, following a 2-year research fellowship in the Laboratory of Biochemistry in the National Heart Institute at NIH. He has been at UCLA for the entirety of his working career. Deborah Hook is a doctoral candidate at the University of California, Davis, within the graduate group of nutritional biology. Deborah's focus is in the area of clinical research with respects to measuring diet, using technology to measure diet, and understanding the nutritional needs of patients with inborn errors of metabolism. She is a pediatric dietitian with 30 years of experience.
Okay. So first of all, I want to really thank Robin and Michael and the Connecting Families UCD Foundation for putting this on tonight. This is just a terrific way of communicating what's going on in the world and all the major advances that are occurring, not only for OTC deficiency, but in many, many rare diseases that have been ignored or overlooked for generations. And so I think we're entering a very exciting time and era for treatment of rare conditions. Tonight's presentation is focused on mRNA approaches to treating OTC deficiency because that's our program, but it's really it's going to be a little more general than that, and I hope to cover a lot of different items with my colleagues on the panel. So first of all, we're going to open up with let me tell you the agenda. Dr. Cederbaum is going to review OTC deficiency treatment and unmet needs. And then we're going to review some of the novel treatment approaches for treating the root cause of OTC deficiency. A little bit of background also on how new drugs are developed, our approach at Arcturus, in other words, mRNA therapy, and also kind of understanding why clinical trials are important to get these new therapies advanced to the market so that people can actually use them and benefit from them. And then finally, Debbie's going to talk about a dietary update and some of the considerations, although I think we agree that her talk could go for the whole time. And so I hope there's an opportunity to ask questions at the end and get more engaged, because I hope that we can do this again sometime in the not too distant future. So without further ado, I'm going to introduce Dr. Cedarbaum to kick us off for review of OTC deficiency.
Hi, everybody. I'm Steve Cedarbaum. As Robin told you, I've been working and caring for patients with urea cycle disorders for 50 years. We hardly knew anything about urea cycle disorders when I started. My job here, however, is really to make sure we're all on the same page and to set the table and background for what David has to say and what he'll describe to you about new therapies. And I'm going to describe why we need these new therapies. Next slide, David. This is a very busy slide, but it's meant to make one point. And that is that the body produces a lot of ammonia. And we've got to get rid of it because it's very highly toxic. And the way we get rid of it is to ship it to the liver. And you can see all those arrows on the left panel aiming ammonia produced elsewhere to the liver. And the liver produces this compound urea that we get rid of in the urine. Well, we produce a lot of urea, which means we make a lot of ammonia, and we've got to get rid of that ammonia. The panel on the right tells you what happens when the urea cycle is not functioning. Ammonia builds up, other products from ammonia like glutamine build up, and they intoxicate multiple organs. And that's really what we have to get rid of, and that's our challenge. Next slide, please. This slide is not meant to intimidate you. It's a urea cycle. It consists of 6 major enzymes, and it really has the machinery to process ammonia and to get rid of it as urea, which you can see in the upper left-hand corner. Ammonia is in red on the top of the screen, and that's really the thing we have to get rid of. Next slide, please, David. In the middle there, you can see a big red X. And we've all learned about life that big red X's are not good. And what you also can see, and I used ornithine transcarbamylase, the enzyme that David's going to talk about, as an illustration. But in fact, you can see that the size of the arrow is diminished, and it's meant to convey the fact that the enzyme is either missing or it's much diminished in activity. And if you look up to the right a little bit toward the top, you see the ammonia in red is much larger than it was. There's an arrow pointing to it. The orotic acid down on the lower right is increased, but that's not important for this discussion tonight. And I'll talk a little bit about current treatments in a moment. Next slide, please. The best way to talk about these conditions and to describe them to you to get us on the same page is to describe a patient. And this patient is AS. Initials don't matter. They could be fictional. The pregnancy and delivery were normal. At 40 hours of age, the young man just was spitting up. At 65 hours, his cry was decreased. He had a diminished response to pain. Muscle tone was increased. He wasn't sucking well, and he wasn't maintaining his body temperature. By 84 hours, things were really looking quite grim. Seizures had occurred. He was in coma and he had lost his muscle tone. He was transferred to UCLA at 6 days of age with an ammonia of 1900. And you all know ammonia should be under 75 or 80, depending on the units that are used. So this was obviously a very grim situation. And he turned out to have ornithine transcarbamylase deficiency. Next slide, please. This slide conveys the idea that not all patients whom we have are that ill in the first days of life. And this can occur for a number of reasons. One is that the enzyme deficiency is less complete. Therefore, you saw the skinny arrow that is meant to convey that idea. But these individuals may have intermittent acute episodes. Some may be more severe than others. These individuals tend to eat a self-selected low-protein diet, just self-selected vegetarian diet. They may have a history of intermittent behavior or psychiatric illness. One could be sleepwalking, and you can imagine. So somebody with very erratic behavior. And there may be a family history of disorders like this that have never been explained. And depending on the urea cycle disorder that you're thinking of, and we're not thinking of OTC does not have consanguinity or cousin marriages, but others might. Next slide, please, David. This is a list of symptoms of acute hyperammonemia or mild hyperammonemia. I'm not going to go down the list. Just remember, if you're interested in the list of symptoms, this is going to be available to you online, and you can look at that at your leisure. So we'll move on. Now, what are the treatments generically for disorders of the urea cycle? One is to remove ammonia, and the other is to reduce ammonia production. And we'll briefly talk about each of these. Next slide, David. This is a grim-looking slide. You can see in the upper right-hand corner that the patient is not a pediatric patient, but they could well be. And this machine is meant to tell you that it's never good to have to be treated with a machine like this. So when you see it, it's not a happy situation. And what this is a dialysis, or in this instance, a variant of dialysis, hemofiltration. It means it's got to get ammonia out of the body. And it does that by really using dialysis or an artificial kidney. Next slide, please, David. So what are the treatments that we have? We can get rid of ammonia, and that is to basically use dialysis, or if the situation is more mild, use hydration and lots of calories to have the body try to shut down its production and to minimize it. But how about stopping or limiting ammonia production? Well, one source of ammonia is dietary proteins. So you want to have a diet that's much limited in protein. And Deb, if she has time at the end, is going to talk about this a little bit. But most of you who have someone in your family or yourselves have the disorder will know exactly what I'm talking about. The other is to provide enough calories so that you don't break down your own body's protein. And of course, we do have to encourage the body to use proteins that are floating around and that might be broken down to ammonia by building up a new protein in the body. And we have to add some essential nutrients to that. And again, Deb will talk about that at the end of the program. Next slide, please. We also have an ability to kind of divert ammonia from the urea cycle, and that's using scavengers. And what scavengers do is pick up amino acids that have ammonia in them and that would ordinarily break down to ammonia and get rid of them as other harmless compounds. And so we give sodium benzoate, sodium phenylbutyrate, and many of you are familiar with these compounds. And we don't have to make urea because we're diverting ammonia around. It's like digging a channel behind a dam in a river so that the overflow can get through. And it's similar to that. Next slide, please. We go back to the original urea cycle slide. When we're treating a stable patient, a patient who has no fever, who is not sick, and who's not stealing food from the refrigerator or otherwise not adhering to the diet, we can control ammonia, but then the patient gets sick or gets born. When you get born, you're breaking down a lot of protein. You can well imagine. It's no piece of cake coming down the birth control. It's hard on the mother, but it's also hard on the baby that's being born. So what we can't control is this sudden breakdown of protein that would otherwise go into the normal urea cycle and not raise ammonia. But if you're in this delicate balance, you can't control it. And that's why we need better stuff. Well, what do we have now? We have liver transplant, which you know is a really big deal. There aren't enough livers. There are complications to it. True, we're getting better and better, but it's still not a panacea or a cure-all. There are some therapies that are being tried, like gene therapy or mRNA therapy. And this is what the lead into David is. David is going to talk about these. Each of them have the potential for being able to handle this sudden explosion of ammonia, but he's going to present it to you. Thank you.
Thank you, Dr. Cedarbaum. Let's see if I can, there we go. So, my section is going to cover quite a few topics, and part of it is going to be mRNA. The focus is mRNA, but I really want people to understand the difference between mRNA, gene therapy, and some of the other approaches that are coming down the pike. So a couple of years ago, I would probably be challenged to find many people that knew the term mRNA. But with the onset of the COVID pandemic, everybody knows the word mRNA, or at least the term mRNA, but I still wonder how many people understand how it works. So part of my discussion is going to be focused on how gene therapy works, how mRNA therapy works, and why Arcturus is taking the approach we are and how far we've come. So to me, this is a very exciting thing. I'm going to start off with just introducing the company that I work for, Arcturus Therapeutics in San Diego, though I live in the Chicago area. So I'm in my basement freezing right now. That's why I have a jacket on. You thought I dressed up for you, I bet. And I did. So Arcturus has been around for about 9 years, and it was founded as a platform company, meaning it was a platform for the development of mRNA therapies using lipid nanoparticle technology that was proprietary to the company. And so it really started with a focus on rare diseases, and OTC deficiency was our very first clinical program, but there have been a number of rare conditions that have been looked at and targeted in preclinical stages. But then a couple of years ago, something weird happened to the world, and it was discovered that mRNA is just a really fascinating and efficient way of delivering vaccines. So everybody heard about mRNA through the Pfizer and Moderna efforts, and know that terminology. And indeed, Arcturus also has a campaign to develop COVID vaccine and other vaccines in the future. And indeed, recently just filed for emergency use authorization in Southeast Asia. So very exciting time for our company. It's not the reason I joined, though. I joined this company because I really find it fascinating to be able to target the cause of disease instead of the symptoms of disease. I think Robin in her introduction said I'd been 25 years in the clinic. It was actually more like over 30 years in the clinic, treating kids with various maladies. I was trained as a pediatric subspecialist. And after a while, it really kind of gets frustrating treating symptoms, and never being able to treat the cause or so-called cure something. And so to me, this was a fascinating and just a wonderful opportunity to try to bring life-changing therapies to people with rare diseases. So I'm going to just now talk about what is DNA, what is mRNA, and how they work. So our genes are made up of DNA. I think you've heard the expression, it's not in my DNA, or it must be in my DNA. Those are usually behaviors they're talking about, but our genes, our genetic makeup is DNA. And genes are the recipes or the templates for making our body's components. So the color of our hair, the color of our eyes, whether left-handed or right-handed, several things. All of our body tissues are determined by genes made up of DNA. So this is the code, or the Bible, essentially, for how our bodies are made, our body components are made. However, DNA is encoded, so it has to be translated for the machinery in the cells to make all these things. And that's the function of messenger RNA or mRNA. What it does is it's like the Dick Tracy decoder ring, which probably many of you are too young to remember. But basically, it takes the instructions from the genes, from the DNA, and transcribes it into messenger RNA, which then can transmit that recipe to the cell's manufacturing line, which is also called the ribosome. And then the manufacturing line follows the mRNA's instructions and assembles amino acids from the cell to make the final product, which in this case is OTC enzyme. In other cases, it's a different kind of protein. Okay, so that's all fine and good. And then a cartoon of this may make it a little bit clearer. So this is a cartoon of a cell. It's a very big cell, but in the middle of the cell is this egg-like looking thing, which is called the nucleus. And the nucleus is a safe harbor for all of our genes. Our DNA is kept in the nucleus. And when it's time to make a certain product for the body, so say we need to make melanin because we're getting too much sun exposure and we need to darken our skin to protect against the sun, then an mRNA is formed to decode the DNA, and that's called transcription. And once this mRNA is formed, it can leave the nucleus and go out to the manufacturing line, which is called the ribosome. And what this ribosome does is it reads this strip of DNA, and as it reads it, it gathers the ingredients for making the protein that the mRNA codes for. So these little purple things are amino acids that the ribosome is assembling per the instructions of the mRNA. So very easy. Now, the one thing to remember, though, is that DNA is permanent. So that what that means is that your genes are there always. And when this cell divides, for example, a duplicate copy is made and it goes into the new cell. Whereas mRNA degrades after a few uses. So mRNA is not forever, it's transient, and DNA is forever. So an important concept to keep in mind. And just in case this isn't enough to sink it in your memory, I have an analogy. I'm a pediatrician at heart, so analogies are part of our DNA. Sorry for the pun. But imagine that your Coca-Cola. Coca-Cola has had a secret recipe for Coke for the last 100+ years. And they keep this safely locked away in a vault somewhere so that nobody really can get it. So this is kind of like somebody's DNA, okay? It's locked away in the nucleus, in this case in a vault, and the instructions are encrypted so that not everybody can read them, okay? So somebody has to go in there and decode that thing. And so now you have a top secret Coca-Cola recipe that's been decoded, but it's written in disappearing ink. So this piece of paper cannot get outside the company and be used by anybody else. It has to be used right away. So that's transmitted to our drink master, who assembles the ingredients from the shelf and makes the final product, the Coca-Cola. Okay. I don't know which of these you're going to remember, but I'm a Pepsi guy, so it's not the right analogy for me, but I thought it was clever. What happens though if something happens to that recipe, that secret recipe that's been in the vault for 100 years? What happens if there was a mistake in it? And so if there's a mistake in your DNA or some kind of a change, that's called a mutation, and it can change the instructions on how to make your final product. And if that happens, of course, if the original recipe is faulty, then the transcribed mRNA or written recipe is also going to be faulty. And then the assembly line is going to have bad instructions. So one of a couple things can happen. Either the drink master will say, you know, I don't understand this at all, and I'm just not going to do anything. Or he'll assemble things according to the instructions and come out with a product that's not exactly right. So in the instance of Coke, it's kind of like if you substituted 3 cups of salt for 3 cups of sugar, you'll end up with something that's kind of brown and fizzy, but it's not going to taste very good and nobody's going to buy it. And so the ways we have to think about ways of fixing mutations, and over the last couple of decades, people have been really thinking about how to approach this, and the research and technology has developed in such a fashion that now in recent years, there are multiple diseases that are being addressed by both of these strategies. So there's a couple of ways of looking at this. One is gene therapy, which would be replacing the DNA or replacing the gene with a normal copy. How is that done? Well, in most circumstances, not all, but in most circumstances, you can take the gene, not you, but a company can take the gene and encase it into a virus particle. In this case, AAV is the one that's very commonly used now. It's adeno-associated virus. And it incorporates this into a virus, and then several of these are injected into the body, and they make their way to the organ of interest. And of course, viruses have been around for a long, long time, hundreds of thousands of years, and humans have been around for a long, long time. So what a virus is meant to do ordinarily is to infect a cell by breaching its membrane, going to the nucleus and breaching that membrane, and then making lots of copies of its own DNA so that it can replicate and cause disease, or at least survive, right? In the case of gene therapy, the virus is inactivated so that it can't do that. But the shell remains the same so that it still detects the cell membrane in the same way, it still goes to the nucleus in the same way, and it releases the DNA, which is then transcribed to the normal mRNA, which is then made into OTC enzyme, okay? So this is a replacement therapy. It's, again, it's a gene. So in the best of circumstances, it would be permanent and be replicated along with the rest of the DNA that the host has. So that's gene therapy, replacing the gene. So what is mRNA therapy? Well, mRNA, as you know, is the messenger between the gene and the manufacturing plant. And so you can make a normal copy of mRNA, and in ordinary circumstances, it's encased in a lipid nanoparticle, or little bits of lipids or fats that form a shell around the mRNA to protect it. and it's able to get into the cell, but not into the nucleus. So once it gets in the cell, the mRNA is released. And once it's released, it can go to that ribosome and start cranking out the enzyme that you need. But it disappears, it's not permanent, so it needs to be replenished over and over. So I'm just going to show you briefly a chart with gene therapy on the left and what we call lunar mRNA technology on the right. Lunar is an acronym, but there's no use of learning what it is because it's changed. So, but basically it's just little bits of lipid. And so let's go down some of these categories and see what happens. Can one form antibodies against the treatment? Well, for viruses, yes, obviously, because we make antibodies when we get infections so that we don't get that infection again. And so some people have pre-existing antibodies to AAV, and one would think that that may decrease the ability for the new gene to get into cells because the particle's being attacked before it can get to the cells by the antibodies. And then sometimes the antibodies form after the dose of gene therapy, which can cause problems for redosing. However, on the mRNA side, there's really very low chance of an antibody forming. It's usually going to be against one of the lipid components, and that's not been shown to reduce your effectiveness of the mRNA. So what about repeated treatment? Well, in the case of gene therapy, no. First of all, in best of circumstances, it should be permanent. So that would be a good thing because you only require one dose. But also, if you did need another dose because the first one didn't take, for example, or if you had side effects, repeated dosing is not possible because of the antibody issue. There are people working on that issue to make it possible to do in the future, but we're not quite there yet. Repeated treatment obviously is possible and beneficial for mRNA therapy because you just need to replenish the mRNA so that the protein can be made again. What about the risk of adverse immune responses? Well, I think I mentioned that, you know, we've had this coexistent with viruses for a long time. And so there is a relatively high incidence of adverse immune responses that people can have to gene therapy, whereas that risk is relatively low with mRNA. Stability, I've already said, the DNA is permanent, mRNA is transient. What about our control over how much protein is made? Well, the control is very low with gene therapy because once it's in there, it's done, and you really can't control the cell's own processes of transcribing it into mRNA. Whereas in mRNA therapy, the control is pretty high because you just adjust the next dose if you need to. Then what about off-target effects? This is called risk of genomic DNA modification, which is an off-target effect. There are some viral vectors that are known to integrate into the genes of the host. And so lentiviruses and retroviruses are two of those. AAV viruses are not as apt to do that, so it's rarer, but there is a chance still of incorporation into the host's own genome, which can disrupt things. It can even cause cancer in some cases, and there are some animal models that show evidence of developing tumors when given these kinds of viral vector gene therapies, including AAV. Whereas that is not a risk with mRNA. It doesn't touch your genes. It doesn't go into the nucleus, as I mentioned. So there's really no effect on your genes. You're not going to become sterile. You're not going to become left-handed if you're right-handed or vice versa. It just doesn't do that. And then finally, the potential to treat children, the age potential of patients. For gene therapy, while you could treat children, but children are still growing. And so as long as the target organ genes are still dividing and growing, it kind of dilutes the effect of the original treatment. And so until the technology exists so that you can retreat with gene therapy, this is not quite as feasible right now with children. Whereas with mRNA, there's no restriction on age. you should be able to dose mRNA in any age group. So those are some of the differences between gene therapy and mRNA. The bottom line is, under great circumstances, gene therapy is forever, and that's good because it's convenient to get one dose of something and then never need it again. But it's bad if you have a side effect from it, because then you can't get rid of it. So on the safety side, not as good. On the efficacy side, great. With mRNA technology, the bad news is that it's the good and bad news is that it needs repeat dosing. But if there is a side effect, you just can hold the doses or lower the doses and get around that. It's not forever because the mRNA disappears. So mRNA has the upper hand on the safety side. And on the efficacy side, potentially it's just as good. It just needs to be replenished. I'm just going to talk a little bit about that lipid nanoparticle technology. The lipid nanoparticle, this looks like a science fiction picture from, one of those space odysseys. But basically what this represents is these green spirals in the middle of this complex thing are the mRNA, the mRNA molecules, okay? But it's mixed in with all these other things, and all these other things are basically just little bits of fat. And if you look closely, this outside membrane of the lipid nanoparticle looks a heck of a lot like the membranes of our cells. And it's because it incorporates the same ingredients that our cells have. These are phospholipids. There's cholesterol inside to help stabilize everything. There's PEG, polyethylene glycol, that helps stability as well. And then there's our proprietary lipid, which I can't mention by name publicly, which helps break this whole thing apart once it's inside the cell to release the mRNA. But what this does, all this stuff does, is it protects the mRNA from being broken down in the bloodstream before it hits the target, because in the bloodstream, there are all these enzymes that break down RNA so it doesn't work. It's RNA in the bloodstream is considered an invader. And so this protects the mRNA from degradation. And it's kind of like if you have your $500 bottle of Chateau Lafite Rothschild that you want to send to Uncle Jake in Minnesota, and you live in Seattle, you don't just put the bottle in the mailbox and expect it to get there, right? Because first of all, it's going to break, and second of all, it doesn't know where it's going. So you need some kind of packaging, like bubble wrap, or in this case, these fancy cardboard thingies with some outer protection as well, and a label so that it's protected from breakage and it knows where it's going. And this is why you can tinker with these ingredients of the lipid nanoparticle to actually target them to different organs in the body. It's really cool science. All right, so now I'm going to shift a little bit to drug development and talk very briefly about it, because this could be its own talk. But really, people don't understand a lot about drug development. Drug development takes a long time. Developing a new medicine is a very rigorous, lengthy, expensive, and complex kind of endeavor. The average development time is around 12 years. Which is probably why a lot of people were suspect of the mRNA vaccines, because it only took, a year or so to develop those. The problem is that they don't realize that a lot of the research and development took place 10 years before that to get it to a place where you could just plug in a new mRNA and go. And that's the beauty of the mRNA technology for vaccines. But anyway, normally it takes about 10 to 12 years to develop a new drug. And the research and development costs can be anywhere from a few $100 million to almost $3 billion per drug. And the reason for this is that there's numerous steps of development. You start with figuring out what your target is and looking at the physiology of what you're trying to correct. And then you can go through hundreds, thousands, or hundreds of thousands of compounds to try to find one that hits the target the way you want it to. And once you optimize that, then you go through preclinical testing, which is where you can look at cell models or animal models and see if it works, and then fine tune it a little bit more there. And also look for safety and toxicity in animals. And finally, when you're through all of that, which is about 5 or 6 steps and takes 2 or 3 years, then you're ready to go into the clinic, which I'll talk about later, and see how it works in humans. The nice thing is that the advances in technology and having a better understanding of the diseases that we're trying to treat is really bringing in a new era of drug development. Because now, not only can we target the root cause of diseases that were thought to be untreatable in the past, but we can develop treatments for diseases that have only a few hundred or a few thousand patients in the world with much more ease than we could before. So the issue is that these new technologies come with new challenges, and it really requires a lot of collaboration between all the stakeholders, including the industry, the companies, regulators, physicians, and patients. So it's really, really important that everyone works together in this drug development process to bring new therapies to be used. I promised I'd go through the phases of clinical trials, and this is not very technical, very high level. Basically, there's three phases in clinical trials and clinical drug development. The first phase is to figure out if the drug is safe. And oftentimes this is done starting with a single dose and then going to multiple doses in healthy volunteers. But in some cases, like for cancer therapeutics, patients are used instead. And for gene therapy, almost always patients are used because there's absolutely no benefit to healthy volunteers to get a gene that they already have a normal copy of. So it's safety, safety, safety for phase 1 trials. They're usually anywhere from 60 to 100 healthy volunteers that are involved in phase 1, sometimes less, sometimes more. But that kind of tells you the range that you're allowed to operate in for doses. Then you go to a phase 2 trial, which involves patients almost always. And what you're doing in a phase 2 trial is looking for safety over several doses in patients, but usually for a short term. If you have a chronic therapy, you might take a 2-week, 6-week, or 3-month period of time to see if it's safe for that period of time. And then you really want to know if it works. This is where you get your proof of concept. And you try to figure out what the best dose is, because you usually are testing several different doses in phase 2 to figure out what the best one is to take forward to the last phase, which is phase 3. And phase 3 trials are longer and larger. They're bigger studies. So for example, for the vaccine trials, it's 20 or 30,000 people. But for, say, something like a hypertension drug, it might be 1,000 or 2,000. For rare diseases, it can be under 100. But the duration of treatment is longer. For a chronic therapy, it can be 6 months to a year and sometimes longer to determine the long-term safety and the efficacy, whether it works or not, and then determine what the benefit-risk ratio is. Is this a drug with lots of side effects, but the benefit is so huge it's still worthwhile going forward? Or is it something that has a little bit of benefit and a lot of risk and it's not worthwhile taking it forward? But I will tell you that most drugs fail in phase 2, not in phase 3, because by the time you get to phase 3, a lot of the risk has been taken out already. So that's clinical trial development. There is something called a phase 4 trial that can occur after the drug is launched, but I'm not going to talk about that in the context of tonight. So now for our program. Our program is called LUNAR OTC. LUNAR is the abbreviation for the lipid nanoparticle technology that we have. And this drug is also known as ARCT-810. I don't think it's the 810th compound that we came up with, but it got this label. And this is the mRNA replacement therapy for OTC deficiency. So just briefly, this should be familiar. The upper left-hand corner is a cartoon representation of the mRNA and all its parts. I'm not going to go through these technically, but basically you you can modify certain regions of the mRNA to make it much more efficient and produce more protein than say a wild type or a normal person would have. So you can actually optimize what so-called normal mRNA is. And we have done so. We have 3 little changes that make it more efficiently translated in the ribosome. And it also makes its way to the part of the cell that it needs to work in much more efficiently. And then the other part is this lipid nanoparticle, which is, it looks like one of Elton John's suits in the 1980s. But basically, it's a lipid nanoparticle technology protecting it from being broken down. So ARC-10 was developed and it was tested in a mouse model of OTC deficiency called the sparse for ash mouse model. I'm not going to go into detail here, but basically these mice were treated with the drug that we developed, and they were challenged either with ammonia or high protein diets. And what it showed is a marked improvement in the urea cycle function, as well as a marked improvement in survival. So the animal models really told the story about how well this worked. And it also kind of defined the dose for the minimum therapeutic range, which is 0.1 milligram per kilogram in this case. So then we got into humans. In 2020, we did a single ascending dose study in healthy volunteers. Remember, phase 1 is healthy volunteers oftentimes. So we had 30 healthy volunteers, 20 of whom got ARC-10 and 10 of whom got placebo. And basically you start at a very low dose and then you increase it little by little until you get to the top dose. All of the doses that we tested were in the proposed therapeutic range. And what we found was that it was very safe. Basically, this was one of the cleanest, the cleanest phase 1 studies that I've seen in my career. Basically, it was generally safe and well tolerated. The adverse events or side effects were very mild. There were some in placebo patients as well. There were no changes in laboratory values, physical exams, EKGs, biomarkers, cytokines, nothing. The only thing that happened was there was an infusion reaction in the first person that got the highest dose. So an infusion reaction is when the body has a system for foreign invaders called the complement system. And what happens if you give this too fast, for example, is that it recognizes an invader and you have a reaction that can result in symptoms like headache, back pain, abdominal discomfort, nausea, things like that. So just kind of non-specific things. You can even get a little bit of a fever and it goes away. So the very first patient had that. And then the infusion protocol was changed to do a little bit slower for the first 15 minutes and followed by the normal rate. And after we changed that infusion protocol, no one else had an infusion reaction. And the nice thing about this whole thing is that we did not have to use steroids as a pre-medication before giving this drug. You'll find with a lot of genetic therapies and LNP therapies, people are giving steroids as pre-treatment. And we know that in OTC deficiency, we do not want to give steroids because it can help breakdown of muscle and cause high ammonia levels, which is exactly what we're trying to prevent. So that was really, really good news. Next is a similar study in people with OTC deficiency. And obviously still the objective, it's still a phase 1 study, it's a single dose, is safety, safety, safety. It's really looking at any safety issues because this is still a relatively new technology. And as a side, we're also looking for improvement in urea cycle function because that's our objective eventually, is to treat the disease. The details are that we now have 8 open sites across the US, and there is a 4-week screening and diet stabilization period, which Deb might touch upon, where people are doing diet diaries and keeping track of what they're eating and trying to maintain about the same amount of protein and calorie intake over a large span of time. And then they're given a single dose of the ARCT-810 or placebo by vein intravenously, and there's a 4-week follow-up period. So a very straightforward 4-week screening period, get 1 dose and a 4-week follow-up period. And in the meantime, we're measuring all kinds of stuff, including urea cycle function and so forth. The eligibility for the trial is men and women 18 years and over with OTC deficiency who have relatively mild and stable disease. So that means an ammonia level less than 100, good cognitive function to allow them to participate and know what they're doing. And so this actually includes women who are carriers or heterozygotes for OTC deficiency. Now you might say, well, what kind of benefit are they going to get from a study like this? And the answer is actually none. To participate in this study is one of the most altruistic things someone could do, because there is no personal benefit for this. It's really knowing that you're participating in the process, and this is the early process, to advance a novel therapy further along and try to get it approved for use in everybody with OTC deficiency. So that's the key. That's the key. Now we're still looking for improvement in urea cycle, but because the people that are in the study are relatively mild, we may or may not see that. We may or may not see that. So we're hoping by the end of this year to wrap this study up. Last year, when I first met Robin and Michael, we were desperate to get people into this thing. COVID was raising its ugly head, and sites were shut down, and it was really miserable, to be honest. And I think it was affecting all clinical trials other than those that were involved with COVID. But recently, over the last, I have to say, the last 2 months, things have mushroomed. The interest in this study has just taken off. And right now, it looks like we're going to have enough people to satisfy full enrollment. But it's still really important to talk about these things, to raise your awareness of what's going on, and to be ready for the next step, because it's coming. It's coming. Now, the next trial that we have planned is a phase 2 trial in the UK and Europe, and that'll start this year and go into next year. And this one will be a multiple dose study looking at safety, tolerability, just like anything else, but also to prove that it works. This is going to be really the first study that will help us show that ARCT-810 actually makes a change in the urea cycle function with OTC deficiency. The treatment period, the screening period is the same as the other study I described. And then ARCT-810 is given every 2 weeks for 12 weeks. So 6 doses in total. And then there's a follow-up period for 12 more weeks. In this study, we're doing it a little bit differently. We're gonna enroll younger people down to age 12 with OTC deficiency, and also relatively stable OTC deficiency, but not as stable as the phase 1 trial. So now we're allowing the ammonia level at baseline to be anything. And the only requirement is that there are no more than 1 or 2 hospitalizations in the last year for metabolic decompensation. So it's a little bit different. We're going a little bit younger, a little bit sicker. This study will probably take at least 1.5 years to go through and enroll. We're going to try to enroll 24 subjects, and we'll try to be as efficient as possible. But really, and I just said this, we really want to establish how safe the drug is and how effective it is so that we can move on, because our ultimate goal is to progress to younger kids and people with more severe OTC dysfunction. It's not our objective to treat mild people, because they may be well off enough not to require something like this, but rather to just really get into the people who need it. And you might ask, well, why don't we do that now? And really the answer is that in general, the sicker people tend to be the younger ones, and you really have to test out the safety in older people first. It's just the way drug development works. So that brings me to kind of the final frontier, and that is kind of what I used to call clinical trial challenges. And believe me, last year you could use the word challenge, and that was probably too mild of a word, but transform this into clinical trial opportunities. And as I said before, this really is a partnership between industry, investigators, patients, regulators, and patient advocacy groups like Connecting Families. It's really important to develop this partnering capacity, and tonight is really a great step in that effort. Now, there are a lot of barriers to efficient clinical research. I mean, everybody would like to enroll a trial as quickly as possible and get your answer. But the fact is that doesn't happen too often. It did with COVID vaccines, believe me, but ordinarily it does not happen. And so patients have barriers. I'll talk about that a little later. Investigators and sites have a complaint list you cannot believe, because all these folks kind of live and breathe in an academic institution, and institutions mean there's a lot of rules and things that may or may not be effective in conducting an efficient clinical trial. And so we have to get through those barriers as well. And then there are regulatory barriers that I'm not going to talk about tonight either. But again, probably the biggest barrier over the last 2 years has been COVID. There have been site closures. There have been loss of personnel at some sites. A lot of sites just shut down any research that didn't have anything to do with COVID. And now I see the sun shining again, and I'm seeing that a lot of trials are opening up, including ours, full blast. And so I think for a while anyway, we'll have the green light to proceed. I'm really happy about that for everybody. So how can sponsors help? Well, really what we can do is make the protocol reasonable. So we should have eligibility criteria that are not too restrictive, try to reduce the number of study visits so it's not too invasive for the family to follow up with, and also limit the amount of assessments we need to just the ones that are needed, and not some ancillary ones that might solve some problems for you. Those can be answered in the future. But really importantly is we need feedback from you. We need feedback from patient groups, from patients, from physicians who treat the patients, to really make sure that we're understanding understanding what we're trying to do, meaning understanding the end game. What are we trying to treat, and also the steps along the way? What can we do to help along the way? Well, how can patients help? Well, I was conducting clinical trials as a clinician for over 25 years, and encouraged, one of the things I did in all my clinics was to discuss the research in the field with my patients at almost every visit. And I think that's really important to develop a culture of research in your clinics. So if you don't get that from your physicians, I would start asking, hey, what's going on, for OTC deficiency? What's going on for whatever else that you may have? And just get an idea of what's out there and what's coming, because believe me, things are starting to pick up a lot, and it's very exciting. But there are a lot of reasons why people don't participate in clinical research. And, it ranges from heavy work and school schedules, which everybody understands, transportation issues, fear of the unknown, fear of needles, or just the thought of being a guinea pig. So there are a lot of reasons not to. So it's up to every individual person to figure out what their motivation is for participating or not participating, and try to weigh priorities, because it really does take a special kind of person with the courage and commitment to overcome barriers. And quite honestly, the rewards of participating can be life-changing. I had several patients after their first experience with a clinical trial. They were emailing me or calling me and saying, when's the next one? When's the next one? When can I participate again? So it's very, very exciting stuff. And then I just wanted to quote someone from over 2,000 years ago, who was Hillel the Elder, who was a rabbinical sage from before Christ's time. And he's well known for a quote that is very pertinent to clinical trials now. In his quote, he said, "If I am not for myself, who shall be for me? If I am not for others, what am I? And if not now, when?" If you have a rare disease, who else is going to step up to do a clinical trial? There aren't that many people to do it. And so if you have the capacity to do it, would this make sense for you? If I am not for others, what am I? I mentioned that the early stage trials may not benefit some people, but eventually will benefit others. And so is that important to you? And of course, if not now, when? As Larry the Cable Guy always said, let's get her done. And so, and I used to live in Florida just down the street from him. So I used to use that phrase a lot. And I would encourage everyone to just kind of think about these things and decide for yourself whether this is something that you might be interested in. And by the way, I don't care if it's our studies or somebody else's studies. A rising tide raises all ships. So, it's really important to think about. So with that, I'd like to thank everyone, especially you all for coming out tonight and listening to us. We're not quite done yet. I'd also like to thank Robin and Michael again and Connecting Families for sponsoring this. This is really a special couple, and their devotion to this community is just unbelievable. And finally, just to mention that there are some resources, and I think you'll recognize some of the people in this picture. This was when we hosted Rare Disease Day last month, and Robin and Michael came down to tell their story, and Dr. Cedarbaum as well. And these are a couple of our scientists that contribute to all this wonderful capabilities that we have now. Wanted to mention our company website. And these two sites are for the clinical trials I mentioned that are ongoing, the phase 1B and the phase 2 study. And finally, if you have any questions at all, you can contact our patient advocacy specialist, who's one of the most enthusiastic and effusive people I've ever met. With that, I thank you, and we'll answer some questions later, but I want to devote the next period of time to Debbie Geary-Hook to talk about dietary issues.
Okay, hello, thank you for having me. And I just, I know that a lot of information has been given to you. It's kind of, you're probably sitting there on overload, a lot of science. And so usually with diet, we'd like to maybe pull that aside at another time and really do a deep dive. Do a deep dive where we do the beginning of diet, for those of you who are new, and then a more advanced diet education for those of you who've been around. But I wanted to stress some kind of important messages that dovetailed in with the presentation. So if you were to participate with this, if let's say in the future this becomes a therapy, there's always a question of, do I get to change my diet? And the big take-home message is you must not change your diet until your doctor and dietitian believe you're ready. And your diet will not change overnight. It'll be changed very, very slowly. This can get frustrating, but it's really important that we know what you're eating, that we can document that so that then we can look at labs and make sure that the changes you're making are still safe. And everyone knows their dietitian. Usually you have her on speed dial. So you'll just be working with her in a little different way, but just as much as normal. Next slide. So just like you do right now, you're going to do after mRNA therapy is keeping things in balance. And some of you have seen this slide of mine before. So that's just calories in equals calories out. Calories are what make us move, makes our body, even when we're sleeping, we're using calories, although we don't think about it. Growth, and then any kind of work that the body wants to do. Well, to do that work, you have to eat food. And most of you are eating a low protein diet, and you're getting your protein in a very special package formula. Next slide. So this is how I think of meals. I kind of break them down, which helps families strategize. So you're not so overwhelmed with what do I put on that plate? What do I buy at the grocery store? So in the center is the meal. So I think of the meal. And the first thing I'm thinking about is foundational foods. And this isn't going to change. So this is your fruits and your vegetables, the things that give you lots of vitamins and minerals and phytochemicals, which we all need in our diet. They're the foods that when you sit down at your table, everyone is eating. And they tend to be very low in protein. So that's where you would start. The next thing you would think down is go down as your switch out foods. And this is where you either do a very low protein product, or as if you have milder disease, you could have, let's say with pasta that has more protein, moving along to regular pasta. And this is a meal that you could have just a few switch outs, but it allows everyone at the table to be eating the same meal. Then you think of your protein source, and where it is right now for most of you is that formula, which everyone, I hear you all want tasty formula, and it's so hard to talk a company into making a formula that just tastes good, doesn't have any more bells and whistles to it. And then again, you have to fill up on calories and fat. Fat gives you calories, but fat also in our diet does many other things. Next slide. So I just have 2 more slides, and I took 2 common meals, our taco meal. And you'll see the arrow going from low to high, and this is going from low protein to high. These are your swappables. So many of you might be doing the low protein tortilla. Some of you might be doing the deep green leaf lettuce leaf instead of something that looks bready. But you move up, and then there's corn tortilla that has a little bit more protein, up to the regular flour tortilla that has even more protein. If you go over to your protein swappable source, you're looking at formula. But as your diet is allowed to be liberalized, you could change that into corn. You could change that into lentils, change that into beans. Notice I didn't say meat, because we still don't know if that is what some of you will be able to do. But again, getting the idea that you're going to slowly move from where you are to something that has more and more natural protein. Next slide. This is my pasta meal, because most of us eat spaghetti. And again, you see that arrow on the left going from low to high, talking about low protein to high. And these are your swappables. So some of you will be doing your low protein pasta, but as your diet liberalizes, you could do spiral zucchini squash, which, believe it or not, has a little bit more protein than your low protein pasta. And then if you have, if you're capable of still handling that protein load, you could move on to regular pasta. But you can see how your menus aren't changing that much. You're just swapping out things in a slow, methodical fashion, very well planned. Swappable number 2 is what I call my protein swappable. You'd go from formula. You might be able to move to your low protein balls, that'd be meatballs, walnut balls, which are made, they're vegetarian, they're made with walnuts, and then maybe even into something that has meat. Again, it won't be a free-for-all, but it might be a little bit of meat, or it might be a little bit of meat mixed with, let's say, walnuts or something else, so you get more of a flavor. And I know that some of you won't even want this. You know, you weren't raised on meat. You find it, you have a huge aversion. So that swappable won't be where you're going to go. You're probably going to spend more of your protein in your diet swapping out into regular starches. And again, your calories, your olive oil, your fruit juice beverage, just trying to meet your calorie needs. And those were the 3 or 4 slides that I had. And again, just trying to bring home the message is, you won't change your diet overnight, and that you'll be working with your dietitian, which you already are, and you're going to make slow, methodical changes that are very well planned. You don't have to really do new menus, so you're not going to go to the store and think, oh my gosh, what am I going to fix for dinner? What am I going to buy? You're going to take your recipes that you have and slowly but surely swap out ingredients. So thank you so much.